Good afternoon, everyone. And yeah, special thanks to the organizers for inviting me here. It's a pleasure to be here and also to get to know some people like you. We've seen our work for quite a while, but didn't actually really meet. We move gears a bit. I think we are kind of doing a backslash. We'll be going a bit more to what we did yesterday. It's far more about biology, and what I actually will do with you is kind of take you to a little journey, also a bit of a historical and maybe also a bit of a philosophical journey through stem cells, especially the hematopoietic stem cells. And let me start out with a quote from Johann Wolfgang von Goethe von Faust I, who says blood is a very special juice, and I can just agree to that and try to give you a little idea about the philosophy behind and also about some of the open questions. Let me start out with this slide. It's rather biological. So what we are actually talking about is a population, a rare population of cells, hematopoietic stem cells. They actually reside mostly in the long bones, but also in other bones, and also in the spleen, especially like in infants, but mostly in the inner regions of the bone. And these cells have unique features. In one place, they maintain their own population over the whole lifetime of an individual, but they also contribute to all types of cells. And you have to keep in mind that each of us is producing about 10 to the 11 blood cells, new blood cells every day. Okay, so there's quite a large turnover in the system, and this only emerges from 10,000 to 100,000 blood stem cells. Okay, so the defining features of these cells is that they do self-renewal, maintain their own population, but they are also multipotent in the sense that they can contribute to many different cell types. All right, let me go back to actually an old paper that Markus Löffler and Chris Potten did in the 1990s. It's actually about crypts, so crypt cells, but many of the features actually apply to other things as well. And one particular thing that they were pointing out, it was very conceptual back at this time. They also got a cover. You can see that here, and I might also like zoom in a bit more. So actually you have the cell cycle here, and then you have like progression, so a differentiation on this downward axis. And the remarkable point in this work was that they actually have this little backward arrow here, which I'm trying to point out. So the idea is that this is not just a unidirectional process that kind of goes down differentiation pathway, but there's actually a balance, at least for these very upper stages, in which certain events can also be reversible. Okay? And this concept was further iterated, but there's actually one more aspect in that paper that I would like to point out, and I'm just quoting this here. They were basically saying, and this is still what we are doing, we're still defining stem cells by their function. Yeah? So we kind of test them in assays, in animals. We try to see how they perform. But the act of testing them, the act of testing their functionality ultimately alters the cells. So it's pretty difficult to like at the same time study a cell which you only can functionally test. So it has kind of this uncertainty principle that we also know from physics. I mean, not to that extent, but it's kind of the same type of feature. If you test a cell, whether it's a stem cell, you ultimately lose it. All right, let's go to another paper that was published a few years later, which was actually by Markus Löffler and Ingo Röder. And they also tried to like define stem cells a bit more in this context. And they actually like came up with a concept that actually like went through a lot of the works that we did over these past 25 years, which is actually the idea that stem cells can behave differently under the influence of different environments. I mean, that's not a totally new fact, and it's not a magic somehow to imagine it, but it was a pretty new concept, and especially the idea that you can actually move around those ideas. So one idea, and you can think of this, called it growth environment A. Back then, you can think of something like a stem cell niche, something that keeps cells quiescent, so they are not dividing, that they keep their stemness behavior. And you have another environment that is more proliferative, and if the cells actually go back and forth, you can get something like a self-organizing population that maintains themselves, but can also, like on the other hand, contribute to differentiation. They actually also like amended the definition, and I'm not going to go into detail. I just like want to point out two things. So the first is like this differentiation behavior, so giving rise to other cell types, while the other one is like conferring to this like self-maintenance or self-renewal behavior. But the important point is that they also said probably this population has a flexibility in doing these things. So it's not one or the other, but it's a flexibility in the use of these options. And back then they also like casted it in a simple agent-based model where you were kind of assuming that you had these different environments. The lower one, which is here in darker gray, is kind of indicating this quiescent state where cells kind of retain their stemness, while in the other compartment they are proliferative and you have like a regulation between them. You can actually also adapt to changing needs. You have something like a self-organizing system. I'm not going too much into detail. I will show you this slide again in two later things where we actually see how this model performs in a real system. Let's go to data. So this was a pretty new concept, this idea of changing stem cells. So this was kind of between these two states, it was kind of a new concept. But then there were also like the first papers arising that kind of caught up on that idea and tried to see how this actually happens in real examples. And there is this landmark paper from Andreas Trumpler aus Heidelberg, where they were actually showing, or they seemed to find indications of what happened here. And let me briefly explain what they did. They actually were feeding BrdU to the drinking water of mice. The point is what happens here is BrdU is integrated into newly synthesized DNA. It replaces thymosin, and it is then kind of stuck to the DNA. So if you feed it, you kind of enrich dividing cells with that label. And if you then take it away and wait, with each cell division event, the label is, so to say, halved and like distributed to the daughter cells. So over time, you will see that the label vanishes. And the faster the cells divide, the quicker you will lose the label. The interesting thing here is the blue population, because this is the stem cell population, the population of which we think that they are the best performing stem cells in that sense. And here the concept is that you see like an initial decline, which is rather fast. But there's also like kind of a slower thing in the end. And this
naive question.
Yes,
so
yes.
You have this BrdU, which you can see, right? When a cell divides, does the amount of BrdU decrease?
No, it's only distributed to the daughters. But you have like a duplication of the DNA, so to say, and if it's kind of like you have like crossovers.
It doesn't decrease. Why does this go down?
Per cell.
Oh, it's per cell. It's so what you do here is you measure it on a per cell level, and it's kind of they have a threshold. So basically, they look for BrdU positive cells. So if they usually it's like 3 divisions, you would say. After 3 divisions, the label is like it's only 1/8 of the initial one, and then it's below the threshold, and you would not count the cell as BrdU positive anymore. Yeah, depends a bit on where you set the threshold. But this is actually what happens. And then the number of BrdU retaining cells, so to say, is lost. Yeah? Okay.
How did they know that they were stem cells for the blue?
Okay. Usually, you're right. I was just saying that this is difficult to define. Usually, you have like marker combinations. So with flow cytometry, you identify cells that have certain typical marker combinations that are at least largely enriched for stem cells. You cannot be sure that each of those cells is a stem cell in a transplantation setting, but 1 out of 3. Okay? Good. So this seems like to have 2 different dynamics in here. And this is also actually how they explain the data. So they wrote like a simple mathematical model, and they argued that this is actually this curve that you can see. So this is the data again, and the curve is kind of a convolution of a fast dividing population and a slow dividing population. Yeah? So 15% of these more or less dormant cells with a very long turnover, and like a larger fraction of cells with a fast turnover. And what you see here is the convolution of those. It's kind of remarkable that the model did not include this switching. So they were actually like just simulating 2 more or less separate populations without actually having the switching, but they were still concluding that, yeah, you see this behavior. So these are like these dormant, deactivated cells, and there's actually a switch between these two states. So the cells could kind of like move around. This was the conclusion that was drawn from that paper. And just to illustrate that, there is a publication which was just published 1 year before that from the same group. And kind of here, this reversibility is still missing. So it was kind of a new idea that obviously there seems to be a back and forth thing back then. All right. We actually caught up on the data because the idea was, can we also like explain this in terms of these reversible features? Yeah. So is there really a way to explain it in that sense? And you can already see from the title of the paper that we kind of termed it in that sense. So maybe these are like not distinct populations, but it's more or less the same thing that comes in two different shades, so to say. And well, we were kind of like using this type of model, yeah? So we were kind of like going ahead using this type of agent-based model where you say cells can come in different flavors, and all the cells that kind of have these extended phases of quiescence, or then, so to say, accounting for this slower turnover. And this is actually what you see. So the data we just looked at is the points in that graph, and you also see that this is kind of now, the red curve is kind of an overlay of this quiescent and deactivated population in here. There was actually similar data generated the year before, now also shown on the log scale. On the log scale, you can really see that initially in the initial population, they all showed this only on the linear scale. You just miss those. You just miss those effects that happen here at these small rates, but also this data can be explained in the same context rather well and easily.
So I'm a bit confused what you're showing there, because kind of the red cells, they just don't divide, and the green ones divide, right?
No. The blue and the
blue one,
yeah.
Doesn't it mean that in the end you have mostly green ones?
No. I'm not saying they are not dividing. The thing is, in the context of this, like if the cells are just like in those activated, so the green ones, you just go from one division to the next. If they, in between, enter this quiescent state, we count them as the blue ones, so to say.
But as time goes on, doesn't the fraction of kind of these cells also change? So because the cell population is growing in this model, or is it?
No. The cell number is more or less constant. So this is like an unperturbed system. You just, you feed the BrdU, but you don't disturb blood. So there's no transplantation or, yeah?
So when you look at stem cells and you look at the BrdU, do we know what those cells really are? Because often it's very hard to distinguish between long-term stem cells, short-term stem cells, progenitor cells. Could this be short-term versus long-term stem cells?
I mean, it's basically a LSK SLAM, so this is the best available population that you usually have. You can do further subdivisions if you look at some other markers, but this is the usual combination that you actually take to identify stem cells. As I'm saying, it's like 1 in 3 might be a stem cell. We don't really know what the others are. They might have like short-term potential. You're right, but this is the best we can have at that stage. It's flow-based. Okay. Good. Then, if you now say, so like we think that the dynamic is a superposition of these actually two dynamics that are going on here, and you can actually now also like map out the distribution of the division times, so to say, and you can actually see that like these the activated cells, they might like divide like every 10 days, every 2 weeks or something. And the quiescent cells, they might stay quiescent for extended periods of time, so 100 days around that. Okay, you can keep the numbers in mind because they reoccur also in other settings, pretty similar. Okay, let me move on to another publication, which actually arrived in 2016, and this is a work actually from Terry Moore's lab in New York back then. And they were actually like doing a pretty similar experiment with a bit of a different technique. In fact, they were using also a marking scheme, but it was not based on BrdU, which is integrated in DNA, but it's actually a system where you have these histone marks, and we discussed it yesterday. It's like proteins on which the DNA is wrapped around. And you can also make systems in which you color those histones, yeah? And so you have like, usually this is drug-based, so if you give the drug, all the histones become green, and if you take the drug away, then also the histones are distributed to the daughter cells, and you have kind of a similar dilution effect going on in the system. And what I observed, I think this is the summary. So if you do this, like initially in young animals, you mark them, and then you actually see that some retain the label and some lose the label over time. But the remarkable thing was when they were looking like later on, like I think this is about after a year, they saw that many cells actually retained some label and stayed there. So there's also a time course here. So this is like basically the experiment. So this is like a fluorescence signal. The gray thing is the background, so to say, what you see in a young uninduced animal. So they've never actually like seen the label, but you still get like some background, so to say, and they just looked what is above it. And they saw that even after 12 months of chase, there are still like a small fraction of cells that actually retain the label. And this is also what you see here. So initially you see this drop, and then the system kind of stabilizes.
Where are those cells measured? Are the animals killed and ground up, or are this blood?
I think this is what you usually do. You sacrifice the animals, you take out from the long bones, you take out the bone marrow, you clean it, and yet then you do like this marker sorting, so you kind of try to identify the best possible stem cell population that you have. Okay. So, but they took like an indication of this level as being an identification that those cells would not divide anymore. So they keep the label and do not divide. So the model behind was that initially you have a label, so you label the system, and then you just wait for division events to go on, and you actually lose the label. We think this is wrong, and there are important parts missing. We think there are two processes missing, and this is actually a background production of the label. So this is a inducible system, so they are always a bit leaky. So even if you don't do anything, over time you will see that there is some background label in the systems. And moreover, there is something like loss by decay. We talk about a fluorescence protein. You can do it in red, you can do it in green, but these are proteins and they have a limited half-time. This is different to the BrdU thing, yeah? You integrate like a thymotine part in the DNA, it sticks there, but this is a protein and it actually decays over time. And if you actually take this into account, the whole interpretation of the experiment changes. We were fighting with Cell for more than a year, and they didn't wanted to see it. It went back and forth, but we finally got it published in Journal of Experimental Medicine. We were actually now including these two processes, and the data analysis was a bit tricky, but I'll try to give you a glimpse of what we actually did back then. This is actually like such a fluorescence profile after 13 weeks. So you kind of label the cells and then you wait for a while. What you see is kind of a distribution. I'll talk about these two peaks in the next step. But we made the assumption that the very right part, yeah, these are the cells that they are the brightest ones. We argue that they did not divide at all or rarely divided. So these are the ones that only lose the label due to decay. And if you actually do this, you can map this out, so you can kind of over time look at this rightmost part of the population and kind of check out how fast does it lose the label. And from these slopes, you can actually kind of estimate this loss by decay rate, okay? And you can actually map it out, and you can see this here. The half-life of this red fluorescence protein is in the order of like 2 weeks or something, yeah? So half of the label is lost even without doing anything. It's a bit better for the GFP, so for the green protein, they stay a bit longer, but they all decay over time. And if you actually map out this parameter, then you get a better idea to map out the other ones. And this is now what we did here. We looked at the other peaks, so to say. How do the other peaks change over time? And from the movement of these labels, you can also like get an idea about how much is actually lost by division. Yeah? And doing these estimates, we can kind of also see that we would argue that these are two populations, and I think it's a bit of the same indication what we discussed before. Some of the cells divide more often, some divide a bit slower, and we can actually end up with similar numbers. So the more proliferative fraction like divides every 2 weeks, and the more slower fraction, the more quiescent fraction would actually only divide every 15 weeks, which is something about 100 days, a similar number that we've seen in the other experiments before. However, this is a strong indication that slowly dividing cells, they might actually get some of this leaky background expression label, and this is why they have it, but it's not an indication that these cells stop dividing. So we still think there's strong indication that this is not that there's still some division going on in this population. All right.
So there are other division markers you could look at, right?
For example?
I'm trying to remember the name of the molecule, K something, you know.
Well, Ki-67, but this only tells you if the cell is currently dividing or not. So it's something that you can use as a marker. You put it on the cell, you see whether it's in S phase, but it doesn't really.
In other words, you can't measure it continuously.
You cannot measure it continuously. The point is also, this is a very rare population. So just looking at a bone fraction, you cannot tell whether this is a stem cell or not. You can only like do it either by functional testing or by this phenotypic marking.
I have a question is that when you play with the cells, so by functional testing, does it change the behavior? So something that may be a stem cell, when you touch it, it back reacts in such a way that. So in other words, is the experiment somehow messing your potential?
For these experiments, we don't think this is the case. For the BRDU, it has some mild toxicity. So it kind of like moves the system a bit, so to say. But for these labeling systems, yeah, not that we see any phenotypes connected to this. And it's also not likely. Okay. All right.
There also are membrane dyes, which will then dilute, and they're not inducible. So I thought there were other ways of testing this besides the one
you can probably do quite some of those things, but you try to do this in an unperturbed setting, which makes it quite a bit more difficult. You can take out cells, you can do transplantations, this is very common, but the transplantation completely changes the dynamic. I mean, that's the idea. You have like a few stem cells, you put them in the system and they totally regenerate everything. But this is not what we want to look for. Okay. All right, we see traces of this back and forth, of this reversible behavior, also if we change a bit. And let me briefly touch the leukemia response. And we've been working on, there's quite some more people here in the room working on chronic myeloid leukemia, and we mentioned the name already before. Chronic myeloid leukemia is a very special leukemia in a sense that it's a very homogeneous disease. So almost all patients have one particular mutation, and this is actually a translocation between chromosomes 9 and 22, and they actually form here this shortened, what we call Philadelphia chromosome, and on this chromosome we have like a fusion oncogene, which is made of these BCR-ABL, so it's kind of like a tyrosine kinase that is constitutive of active. The point is that this mark is unique, so almost all CML patients have it. On top of it, it's measurable. You can do a PCR, and with this PCR, you can pretty well track how much tumor load is in every patient. And even better, you can target it. There are actually drugs designated precisely for BCR-ABL, or for the oncoprotein, so to say, and you have like a detailed drug that goes under these things, which makes this, and on top of it, you have like a clinical community that since 25 years pretty well standardizes and measures these tumor loads over time. So we have thousands of time courses of patients that actually respond to this type of therapy, okay? Which makes it really interesting for modeling people, because this is something pretty reproducible. What actually happens if you treat patients, so this is over a cohort, these are individual patients, but the pattern is pretty consistent. Like initially, again, we see like a strong loss of tumor load. Like in the first year, you almost lose about a factor of 10 to the power of 3, so 1,000-fold reduction in tumor load within the first year. And then you see a slower decline in the long run. And this is also something that you see for individual patients. Yeah? So initially they respond really quickly. This is also what you aim for. And in the long run, it's a slower decline. And actually, we actually also tried to explain this in this context. So if you make some assumption, now you have like a competitive process between normal cells and these Philadelphia positive leukemic cells. And if you now argue that they are targeted by the imatinib, only those, not the others, you can actually describe those time courses pretty well, like with the initial fast decrease and then the slower decrease in the long run, okay? That's a bit of the question, what are the mechanisms behind it? This is actually work that I did together with my colleague Otto, who is also here, and you can already see, we published this in Hematological, like a standard like hematology journal, and I think this is also more the target audience on our side. What Otto actually did, he was kind of reformulating this whole thing that we had in this agent-based model into a simple ODE model. So the idea is that you have these quiescent populations, so they are not really dividing, and we have like this proliferative fraction, and cells can kind of transit between these two states, okay? So the same thing, a bit simpler, so to say, and you can actually translate this into a very simple ODE system. It's a linear system. You can actually solve that also pretty easily. Just as a remark, we kind of like formulated something like an effective toxicity. So this is kind of the proliferation minus the drug effect, or like the other way around. So the drug effect minus the effect of the proliferation. So this is actually the drug effect in the system. And if you actually solve that, you get like a superposition of two exponentials. And now you might recall, also in the data, we see kind of this biphasic type of behaviors, yeah? And this is also what's in this solution in here. The cool thing is that the eigenvalues of this solution can now be expressed in terms of the model parameters. So all these model parameters, all these activation, inactivation rates, like the efficiency of the drug, so to say. And if you actually have that, you can do another step. And I think this was Otto's intuition to see that these parameters actually act on very different scales. So some of them are larger, some of them are smaller. And if you actually employ that, and have a look down here, you can actually reformulate them and identify leading order terms. And you see that one of these exponentials is governed by this toxicity effect, and the other one is actually governed by this transition effect.
I'm very confused. The equations on the lower left and upper right should be identical. Could it be that you exchange the two lines, the upper right? Because the toxicity should affect the dynamics of y, right? Not the dynamics of x, and the q is in the equation for x, or is. Maybe I'm just confused. If I just do the matrix multiplication, I would get the lower left probably only if I exchange the two lines on the upper right. The q should affect the differential equation for y and not the differential equation for x.
On upper right in the matrix.
Yeah.
It's just typos.
Yeah.
Okay, so the lower left is correct, right?
This is the correct thing, so it appears here, you're right. Okay, good. Yeah. Good point. I actually shown this slide quite a couple of times without never really realizing it. So. Good. All right. Let me like give you a visual explanation of what this means. So what we see here, I mean, we have now like more like a downstream population. This is what you would actually measure, but this what you actually measure this downstream population is kind of like a reflection of this proliferative fraction. So what you see with the solution is you see this like biphasic decline. If you map it out on a logarithmic axis, you see it as like straight lines. And you can actually now identify that the first slope is like mainly governed by this cytotoxic effect. And it also makes sense, initially you have many of these tumor cells, and you have a strong cytotoxicity, so you actually eradicate a lot of those cells, and you see this initial decline. The interesting aspect is that the second decline is mainly governed by the rare activation of these quiescent cells. So they are kind of sticking there, and they are only rarely activated over time, which indicates that this second slope is no longer, not to a strong extent, depending on the toxicity, so the amount of drug that you have, but far more by the rare activation of those cells. And this is now actually something that you can really translate into the clinic, because it tells you that in the long run, it's not the amount of drug that you give to the patients. You have to give them a sufficient amount, but it does not necessarily be as high as it's initially expected. And this is also something that we now see more and more in the clinic, that patients respond well. Some of them can stop, but that's another story. But some of them have to stay on the drug. But they can obviously do well with sufficiently reduced doses. So this is actually something that is also very concurrent with the type of model that we did here. All right. Let me come to a final aspect that I would like to share with you, which goes more into HSC heterogeneity. And I'm actually referring to a paper by Katrin Busch from the Rodewald lab. Dominic was also bringing up those slides again, so I might go through that a bit more quickly. What they actually did was they were also inducing a label. So it's similar to what we had with this BrdU thing or with these H2P histones, but they kind of used a different technique. They actually tried to put the label only in stem cells. You can do that if you have a gene that is only expressed in stem cells, and you kind of couple this with this induction system, you can actually induce a label that is only present in the stem cell population. You try to kind of flip something in the genome of the stem cell so that they constitutively express a fluorescence tag, so to say. And the cool thing is that this is then also inherited to the progeny of those cells. Let me show you how this looks in the experimental setting. So if you induce the label, so to say, so they use a yellow fluorescence protein, and if you induce it, you can see that this is the stem cell population. These are the downstream populations here. Initially, if you induce it, you only have the label in the stem cells. What will now happen over time is that the label is kind of distributed to these downstream progenitors. And this is also what you see here. So if you wait for a certain amount of time, you see that the label also enriches in the other populations. And if you now map that out, you have time courses. You see that the stem cell population is considered to be constant. I will come back to this in a second because you can put a question mark there. But what you actually see is you look at the fraction of labeled cells compared to the stem cells in the other compartments. And over time, you would expect that you see the same fraction of labeled cells in the downstream compartment as in the stem cell compartment. And you can fit this, and this is a very simple model that Thomas Höfer and his colleague did in these experiments. And they actually also found that this most quiescent cell population divides only around once in 100 days or something. The more downstream, they divide a bit faster, in the order of 3 weeks or something. All right, all good, very nice paper, very influential, did a lot of good things. They made the assumption that this population is constant, and I've enlarged this. Well, it might not. Actually, there's a follow-up. There is actually a paper which emerged from the same lab. It was put on bioRxiv in 2020. It was never published, or never really published in a peer-reviewed journal. But what actually you can see here is kind of the same thing here, but now they waited longer. So this is kind of more than the lifetime of a mouse. And you can actually see that while initially it may stabilize here around 0.1, so 1%, over time, this starts to increase even more. If you think about this conceptually, this has quite some implication, because the only way to explain this is if you assume a further heterogeneity in this stem cell population, yeah? Conceptually, you would argue if you labeled all the stem cells, so if you labeled this population completely or to a certain level, you would expect that it stays constant. You can only actually explain this if you think of a subpopulation, maybe they call it tip stem cells, so kind of the best of the stem cells on top, so to say. If they are labeled, they might then like even distribute the label within that population. I'm actually showing you a slide that actually emerged from an MD thesis we did a long time ago, and we never actually published, but it's actually kind of trying to do that. So we kind of further subdivided the stem cell compartment, so to say, and now looked how the flux goes through that. And if you assume that only the stem cells, but they are all labeled equally, so in all of those subcompartments, you would get this straight line, yeah, pretty much as was Thomas Höfer was assuming. But if you actually go there and assume that also the label is inhomogeneous in that compartment, so that there are like some very early stages that are actually marked a bit higher, then you actually get to see this type of behavior. You get to see an increase even in this phase.
Because the more, like, less stem-like cells differentiate out, and the concentration of the label increases because the most stem-like cells remain. Is that why?
No, I think I would argue, we would argue that these long-term stem cells are like the self-maintaining population, so they are not lost over time. But because, basically, you would first fill up this compartment until this stage. So these are like only marked to a lesser extent. But if they are all so basically, what happens over time is that this gets a straight line. So everything gets yellow until this point, and you kind of fill up this non-marked, non-tip stem cells, so to say. Yeah. All right. So this is all this labeling perspective, and I try to now like move this in the final final slides towards actually like complementary behaviors that also look at stem cell processes from a different perspective. And we are now actually coming to what we've just also heard in talks before. We have like these single cell analysis type of things, which is not longitudinal in the sense that you track certain processes over time, but that you look at one time point, but at many cells, so to say. And this is actually from a really nice review from Elisa Laurenti and BrdU Göttgens that kind of like summarize the kind of these findings. So this is kind of also this hematopoietic tree that I was showing you. You have like these topmost stem cells, and then they kind of differentiate downwards. This picture changes a bit, so they identified more subpopulations with different potentials. They also saw that there are like some cross-links in these things. But if you actually start to come from a single cell basis, you actually see that these are not distinct spaces. So that cells are not jumping from one compartment to the other, but it appears that they are far more linked. It appears there are like tracks in this landscape. And I mean, intuitively speaking, this seems clear that cells are not really jumping from one to the other aspect. So what we are here doing is more or less like we kind of take certain spots in this differentiation hierarchy, but in the end, it might be far more a continuous representation of the underlying process. Much of this goes back, and I'm just like showing one paper that kind of like gave rise also to this interpretation, which was kind of like just 1 year before this review, which is also like largely done in Heidelberg, where they were actually like laying out human hematopoietic lineage commitment. And they argue it's a continuous process. So the idea is that you actually take different cells, look at them and try to qualify what they are in terms of what they contribute to. And if you map this out in such a two-dimensional space, you have this very large space in these RNA expression states, But you can actually see that there's, so like these sparks, so to say, they correspond to this differentiation pathway, so to say. But if you actually overlay this with like stem cell markers, you see that like stem cells are kind of in the middle here. So they seem to already like pre-express something that is specific for the downstream lineages, but they are kind of like confined here in this region, in this overexpression region. And also it's not a clear distinction between these stages, but it's a continuum. And if you actually map that out, you can see that initially you mark like in the topmost, if you look for certain stem cell markers, you see that they are like kind of concentrated in the middle of this star. And the further you actually go down, you look for particular genes that are like remarkable of certain of these downstream lineages, so to say. You see that you're kind of going down in this trajectories. And you can kind of now put this into these landscape type of things where you have like a large level of heterogeneity here at the topmost level, and then the further you go down, it's kind of more and more restricted. And I have 1 minute. I'm coming to the summary slide, so I kind of like nicely in here. It's a bit of the question whether quiescence and activation are really two distinct states. And it appears that you can explain quite some data with that quite well, but the question is still whether this is like discrete states or whether there's also something like a continuous spectrum of functional activity. And in some sense, this continuous concept can probably much better like reflect functional and phenotypic heterogeneity. And just to make it very short, I think that HSC heterogeneity is a feature and not a mistake. With this, I should mention quite a number of people that contributed here to this work over many years, also the work of the others that I cited. Yeah, thank you.